Okay, lovely students, welcome back to another Anatomy and Physiology 1 lecture for Austin Community College intro students. My name is Matthew Belzer. I'm your instructor. Many of you probably already know that. And this video is the first in a video series you're going to get post-COVID-19 after the spring break, because now these classes have shifted to be in an entirely online format. Now, I got an email recently saying, I think Crash Course does their videos better. Well, of course they do! Crash Course has like 9 people, including like an illustrator, a graphic designer. They brainstorm. They have editing hours. I'm just cranking these out one by one, man, to try and help you out with A&P too. So, yes, of course they do. But we're covering the Common Course objectives established by Austin Community College in order to get our SACS accreditation, and that's what these videos are covering. So we're going to jump right into it, and I know that some of these drag on a bit. Don't try and do it all in one sit. Maybe, take 3 or 4 times at it. 2 hours a day is better than 8 hours in one day. And just like every lecture that you've had in class, what I'm going to do is I'm going to say, hey, we've got a set of learning objectives, and you also have a lecture handout. So have that lecture handout up and available, ready for yourself to follow through the key points as we go through today's lecture. One of the nice things about having an online lecture is that you're able to go back and you're able to look or listen to the material again so you can ingest it. So if you find me going a little bit quick, you can pause it and you can think about the questions that you have on those handouts. But those handouts are walking you through the key points. Now, the key points that we're going to be going over today is the sodium-potassium pump and the electrochemical gradient, which then plays into a concept called membrane potential, which is extraordinarily important. We're going to talk about secondary active transport, the maintenance of water balance, and endocrine sensing, which are some of the things we use the electrochemical gradient for, right? We establish the electrochemical gradient, then we use it to do work, and those are some of the tasks that the electrochemical gradient facilitates. And then we're going to talk about membrane permeability to ions and channels. We're going to talk about what's meant by depolarization and hyperpolarization. And without further ado, there are your learning objectives for this particular lecture, so let's hop into it. Whenever you hear the term membrane potential, what you are talking about is a charge differential across a membrane. In other words, you're talking about the difference in the charge between the outside of the cell, the ECF, and the inside of the cell, or the ICF. Now, if I say resting membrane potential, what I'm saying is that a cell, like a neuron or a skeletal muscle fiber or a cardiac muscle fiber, when it's not firing an action potential, when there isn't an electrical signal passing through it, at rest, when it's just sitting there being a cell, if I say resting membrane potential is always negative in these cells, I'm saying that the inside of the cell always carries a net negative charge comparative to the outside of the cell. So membrane potential is the charge difference across a cell membrane. All living cells have a net negative membrane potential at rest. So the inside of the cell carries a net negative charge comparative to the outside of the cell, which carries a net positive charge. One of the numbers you're going to have to remember for this upcoming exam is that the resting membrane potential, the negative charge inside the cell comparative to outside of the cell for an average or a typical neuron, is around negative 70 millivolts. One way that we denote membrane potential is with capital V, lowercase m. So lowercase m, capital V is a unit of measurement that's measuring electrical charge. It's millivolt. Capital V, lowercase m means membrane potential. So if I say which of the following would be true of the capital V lowercase m of a neuron, what I'm saying is which of the following would be true of the membrane potential for a neuron. And I don't have like a tablet to draw on, or I'd throw that up, but I'm running a little bit shy on time, so I'm just going to get this up for y'all. So we know that membrane potential is the charge differential between the inside and the outside of the cell at rest, right? Biological systems, cells in biological systems carry a net negative membrane potential. Neurons carry a net negative potential of negative 70 millivolts. Now, as a precursor to understanding concepts such as the action potential, we have to review types of transport. So in the first exam, we talked about passive transport. Passive transport doesn't require metabolic energy in the form of ATP. It moves particles with their concentration gradient, In other words, if you see particles moving from areas of high to areas of low concentration, right, those particles are moving with their concentration gradient, and there's no reason to expend energy in order to do that. And the types of passive transport we went over were simple diffusion, osmosis, which is essentially a special word for the diffusion of water, channel-mediated facilitated diffusion, which carries small charged particles such as things like sodium ions, potassium ions, etc., across the cell membrane, and carrier-mediated facilitated diffusion, which carries large charged or polar molecules like glucose and amino acids. So we went over that in exam 1. Now, what we're going to be going over today, right, and the title of this lecture is Primary Active Transport and the Electrochemical Gradient. Whereas passive transport doesn't use metabolic energy in the form of ATP, primary active transport uses ATP directly, meaning that proteins involved in this transport mechanism actually hydrolyze, break down ATP, and use the energy from that to change their shape in order to move particles not with their concentration gradient, but against their concentration gradient. So primary active transport moves particles against their concentration gradient, and that's why we need energy, because we're working against a concentration gradient. It's the only reason you're alive, and the types we're going to look at with respect to active transport are endocytosis, exocytosis, but we're going to draw an especially close focus to the activity of the sodium-potassium pump. Now, secondary active transport also moves solute particles against their concentration gradient, but what it does is it couples the particles that are created during the primary active transport processes such as sodium. It couples the movement of sodium particles to other particles, and it uses the electrochemical gradient to move particles against their concentration gradient. And that'll make a lot more sense when we see the images and things like that of secondary active transport. So when you look at this image, one, you know that it has to be passive transport because these particles are moving with their concentration gradient from areas of high to areas of low concentration. Particles that undergo simple diffusion that are capable of passing directly across the phospholipid bilayer tend to be small nonpolar particles, things like oxygen, carbon dioxide. Particles undergoing channel-mediated facilitated diffusion, remember this is a protein channel right here, tend to be small charged particles like sodium, potassium, calcium, chloride, magnesium. Remember that these channels are proteins, meaning that we have genes, and through the process of transcription, those make mRNA. Through the process of translation, a protein is made, and then that protein is embedded in the surface of the cell membrane. So this is actually knit together by amino acids, right? This structure is formed by amino acids linked together covalently that form in this interesting three-dimensional shape that allows them to conduct ions. This is a protein we're looking at right here. A lot of genetic disorders can affect these channel proteins. They're called channelopathies. So remember also that these channels are specific. So if I say a sodium channel, that channel can conduct sodium and only sodium. If I say a potassium channel, that channel can conduct potassium and only potassium. So channels tend to conduct small polar molecules such as ions, the inorganic ions. Carriers tend to transport larger polar molecules such as sugars and amino acids. Again, if this is a glucose transporter, it's going to bind to and move glucose. But these are still considered passive transport. They're forms of facilitated diffusion because the particles are moving with their concentration gradient. So you don't see the expenditure of ATP here. That's in stark contrast to the different active transport processes that we're going to talk about. Now, our focal point today is going to be on pumps, but I do want to highlight a few active transport processes that I want you to be familiar with. Now, even though you can't see the little protein that's dragging this vesicle to the cell membrane, whenever you have a little, membranous structure within the cell, membrane-bound structure within the cell that's moving substances around, this membrane-bound structure is called a vesicle. Now, when you want to ship that material that's contained within a vesicle, let's say that that's the material in that vesicle is like a neurotransmitter or a hormone or something like that. What ends up happening is those vesicles mobilize, and in order to mobilize those vesicles, it takes energy for the proteins that physically move those vesicles around. So it's considered a form of active transport because we're moving things using energy across the cell membrane. This form of active transport is called exocytosis. So if I say the binding of calcium mobilizes, right, vesicles that then fuse to the membrane and carry out the process of exocytosis. This is what I mean by fusion of the membrane. You have these little sacs called vesicles, and they physically fuse to the plasma membrane, and they release that content into the ECF, where it can be transported to other cells. And it's usually a cell signaling molecule with this class, with what we're going to be talking about, and it can influence the activity of other cells. So this is called exocytosis. But if you hear the mobilization of vesicles and the fusion to the membrane, which facilitates, the answer to that would be exocytosis, because that's what's really happening. Then you have what's called receptor-mediated endocytosis, whereas exocytosis can release large amounts of solute into the extracellular fluid. Endocytosis, and specifically receptor-mediated endocytosis, can bring in large amounts of solute from the extracellular fluid to the intracellular fluid. So one of the ways in which this happens, if you have this thing called an LDL receptor protein, and remember, low-density lipoproteins transport cholesterol around. Now, cholesterol doesn't dissolve directly in the blood. It can't, so it needs a mechanism to physically move around, and that mechanism are these low-density lipoproteins. So if you look at this receptor-mediated endocytosis, what you have are these low-density lipoproteins within this liposome, and they're binding to these receptors in the surface of the cell membrane. When they bind, the cell forms a little vesicle around them. It causes a conformational change in the cell membrane, in which it forms a little vesicle around them, and it brings a ton of that cholesterol into the cell. Receptor-mediated endocytosis, so whereas this is exocytosis, we're releasing large amounts of material out of the cell, receptor-mediated endocytosis is bringing large amounts of material into the cell. Both of those are considered forms of active transport. You don't see the ATP directly, and for this class, just trust me on it, but ATP is being used. Now, the type of primary active transport that we're going to draw a focal point to today is pumps, or ATPases. If I say pump or ATPase, I'm saying the same thing. We're talking about a protein that physically moves solute particles from point A to point B. And the most important protein by a long shot, by an absolute football field, for you to understand coming out of an anatomy and physiology class, is the sodium-potassium pump. It is the most important protein on the face of the planet. And if you don't have a good understanding of the material we've covered up until this point, you may find yourself asking kind of silly questions like, how do sodium and potassium make a pump? I've gotten that question from a student before. Well, sodium and potassium don't make a pump. We have DNA, right? And we have these long strands of DNA, and these functional units of DNA called genes, through the process of transcription, make RNA. And then through the process of translation, that RNA, right, is used to construct a protein, and that protein folds into a three-dimensional shape where it's ultimately shipped and sometimes works with other proteins to form, right, a functional piece of machinery in the cell. So when I talk about the sodium-potassium pump is a protein, right? It's made from amino acids linked together via peptide bonds that fold into a three-dimensional shape. And what this protein does is it functions like a piece of machinery. It's a piece of cellular machinery that physically moves sodium out of the cell and potassium into the cell. I would argue that the sodium-potassium pump is the most important protein in all biological systems. I'm just going to make that argument right now. So you know it's going to be heavily focused on the exam. You know that you're probably going to get a short answer question over the mechanics, just the basic mechanics and the establishment of the electrochemical gradient on the exam. So there are a lot of different ATPases / pumps. There's calcium ATPases, hydrogen ATPases, hydrogen potassium ATPases. What we're really saying is we're talking about pumps. We're talking about primary active transporters that physically bind to things like calcium and move that calcium not with their concentration gradient, but against its concentration gradient, right? The most important of the pumps that we're going to talk about are the ATPases, is the sodium potassium pump. You've probably seen me wear 3 or 4 different shirts that have the sodium potassium pump on it. It's an extraordinarily important protein. Now, when you think about these different pumps, they are pieces of machinery embedded within the cell membrane that physically move solute particles around. So if you hear terms like proton pumps, remember protons are just hydrogen ions. Those are, right, proteins that physically move hydrogen from areas of low to area, or from areas of low to areas of high concentration against their concentration gradient. And notice that ATP is being used. You have hydrogen potassium pumps, which move hydrogen into the lumen of your stomach. Notice that ATP is being used. You don't even need to know what's being physically moved here, right? It's a calcium pump, but what you're looking at is ATP. It's coming in here. That ATP is being hydrolyzed, and then the energy from it is being used to move solute particles, in this case calcium ions, against their concentration gradient. So if you see the use of ATP, you know you're looking at a primary active transport process. Now, what we are looking at here is just a tiny chunk of one cell's membrane with one sodium potassium pump. I once asked a question in one of my classes that was, how many sodium potassium pumps do you think you have in the body? And I was looking for a very rough answer here. And about 15% of my students said 1, right? You find 1 sodium potassium pump, and then they diagram this particular image out. The reason that that's troubling is it represents a very significant lack of understanding of the material leading up to talking about this process. We don't have just one sodium potassium pump. Remember when we were talking about proteins and we were comparing the size of proteins to the size of cells? Proteins are much, much smaller than cells, right? Orders of magnitude smaller than cells. And you can have, in every one of the 200 trillion cells in your body, you can have anywhere from thousands to millions of sodium potassium pumps per cell. 200 trillion cells, you're talking about trillions and trillions of these little pumps using energy to physically move sodium ions and potassium ions in your body. We have tons of them. And there's an argument to be made that this is the most important process in all of biological science. I would make that argument. I mean, some people may retort with some of the plant proteins and some of the, energy, the photosystem proteins, but I would still make the argument that the sodium potassium pump is more important, maybe because I'm a physiologist. So, when you're looking at this is a blob model, essentially, of a protein. Proteins are little pieces of machinery. We have this sodium potassium pump. Here's our ICF, or our intracellular fluid, and here's our ECF, or our extracellular fluid. The first thing I want you to really understand is just the mechanics of the sodium potassium pump. So for every cycle of the sodium potassium pump, 3 sodium ions are moved from the ECF to the ICF. 2 potassium ions are moved from the ECF to the ICF. So I hope I didn't say that wrong. 3 sodium ions are moved from the ICF to the ECF. 2 potassium ions are moved from the ECF to the ICF at the expense of 1 molecule of ATP. So we have 3 sodium ions out of the cell, 2 potassium ions into the cell at the expense of 1 ATP molecule. We know this is primary active transport because ATP is being used directly by that protein, not to move these solute particles with their concentration gradient, but to move these solute particles against their concentration gradient. Now, what does that eventually develop if we activate this sodium potassium pump? Now, let's say you got a question like this on the exam. So you're looking at this cell system. Here's a representation of a cell right here. That black line represents the cell membrane, and we're looking at one representative sodium potassium pump protein embedded within that membrane to draw a point, or to make a point. So we have mitochondria, and mitochondria produce ATP. So this particular process requires energy in the form of ATP because we're not moving solutes with their concentration gradient, we're moving them against their concentration gradient. And let's pretend this cell we're looking at here is a neuron. Neurons have millions of sodium potassium pumps embedded in their membrane because they are electrically active cells. They rely on ionic, what's called disequilibrium. Not equilibrium, but disequilibrium. So let's look at what that means. Now, if I was to ask you to calculate the total charge in the cell, just arbitrary unit, let's say millivolts, but just total charge in the cell, the way you do that is you would count the number of positive charges in the cell. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9. And then you'd count the number of negative charges in the cell. Now I've put P neg for negatively charged protein, but it's important to realize that this could be any anion. So it could be a protein, it could be a phosphate from the breakdown of ATP. This little phosphate carries a negative charge. It could be a chloride ion, could be a number of different ions. But because proteins are fixed and they don't move across the membrane, I focus on those with respect to the fixed charge inside the cell just to emphasize what the sodium-potassium pump is doing. Now, if there's 1, 2, 3, 4, 5, 6, 7, 8, 9 negatively charged particles in the cell, and there's 9 positively charged particles in the cell, this neuron at the moment is neutrally charged. Now let's turn on the sodium-potassium pump and see what happens. So for every cycle of the sodium-potassium pump, 3 sodium ions, 1, 2, 3, they don't go one at a time, they all go one at once, but I'm making a point, are moved from the ICF to the ECF. 2 potassium ions are moved from the ECF to the ICF at the expense of 1 molecule of ATP. Now recalculate that charge internally in the cell. Well, there's still 9 negative charges, but because we moved out 3 positive and only moved back in 2 positive charges, right, there's 1 more negative charge than positive charge. And now that cell takes on, let's say, a negative charge of -1. So with every cycle of the sodium-potassium pump, you're moving out net positive charge. And if you're moving out net positive charge and you have a fixed negative charge in the cell is progressively going to become more negative until it hits a point that we're going to talk about in a later lecture. So if we were to run that again, 3 sodium ions from the ICF to the ECF, 2 potassium ions from the ECF to the ICF at the expense of 1 molecule of ATP, now we only have 7 positive charges in the cell, so it takes on a charge of -2. So when you think about the chemical disequilibrium, meaning the separation of particles, the unequal separation of particles between the outside and the inside of the cell, what maintains that? 2 potassiums in. Now it takes on a charge of -3. That cell was supposed to be bigger, but I don't have enough time to correct it. Right? So when you look at these images and you see that sodium has a much higher concentration outside the cell than inside the cell, that's a consequence of the activity of the sodium-potassium pump. When you see that potassium has a higher concentration inside the cell than outside of the cell, that's a consequence of the activity of the sodium-potassium pump. With every cycle of the sodium-potassium pump, you're moving out net positive charge. And what the sodium-potassium pump really establishes is what's called an electrochemical gradient, right? It plays a role in establishing the electrochemical gradient. So that's why you get that separation when you're looking at the relative concentration of ions in the ECF and the ICF. Why is sodium high? Why does sodium have a higher concentration? It is the activity of the sodium-potassium pump. Remember that the food you eat, right, the energy you extract from food you eat, anywhere from 20 to 40% of that energy is going to this one process. It's an extraordinarily important thing, maintaining this ionic disequilibrium. Not equilibrium, disequilibrium, meaning that we have a differential ionic distribution. Sodium has a higher concentration outside the cell. Potassium has a higher concentration inside the cell. We expend a lot of energy making sure that sodium has a higher concentration outside the cell and potassium has a higher concentration inside the cell. So if you look at the activity of the sodium-potassium pump, whoa, that went pretty darn quick. So the binding of potassium when it's exposed to the external environment, the binding of sodium when it's exposed to the internal environment, 3 sodiums out, 2 potassiums in at the expense of 1 molecule of ATP. What ends up happening is positively charged particles accumulate outside the cell, negatively charged because of those fixed negative charges, those negatively charged particles like these negatively charged proteins accumulate inside the cell, And that's how cells maintain, right, what's called their resting membrane potential. It's one of the, it plays a role in the maintenance of their resting membrane potential. So when you think about why does sodium have a higher concentration outside the cell than inside the cell, and why does potassium have a higher concentration inside the cell than outside the cell, that's all a consequence of the sodium-potassium pump. And as you're going to see as we talk about neurophysiology and muscle physiology, we use that gradient that's created, that what we call electrochemical gradient, to do work. We use it for a ton of different things. So if I say describe the establishment of the electrochemical gradient using the sodium potassium pump as a basis for your answer. One way to do that really effectively is to diagram it out, or so diagram it out, and what you're looking at here is a cell system, and within that cell membrane you have the sodium potassium pump, which is a functional protein embedded. And for every cycle of the sodium potassium pump, sodium ions are moved from the ICF to the ECF. 2 potassium ions are moved from the ECF to the ICF at the expense of 1 molecule of ATP. This is a primary active transport process. We're moving solute particles against their concentration gradient, not with their concentration gradient. Now, due to the activity of this pump and other things that we're going to talk about in just a few minutes, the net charge inside the cell is negative, and the net charge outside the cell is positive. Now, when I talk about the sodium-potassium pump establishing what's called an electrochemical gradient, what I'm saying is the sodium-potassium pump establishes a gradient for sodium and potassium that has both an electrical component and a chemical component. So as sodium starts to accumulate outside the cell, sodium is positively charged. Positively charged ions are attracted to negatively charged, right, ions. Sodium, the energetic forces that promote the movement of sodium, because sodium is positively charged and the inside of the cell is negatively charged, the movement of sodium back into the cell is energetically favorable, meaning that sodium, right, has a force promoting its movement back into the cell, but it can't move directly across the plasma membrane because it's a non-penetrant solute. But there is a force, right, tugging on that sodium, trying to pull it back into the cell. Now, some textbooks talk about sodium wanting to get back into the cell. Sodium can't really want anything, because want is a complex behavioral state that's a construct of interacting neurons and has to do with emergent properties of interactive neurons. So I don't know what that means, but the energetic forces promoting the movement of particles promote the movement of sodium back into the cell if it's given an opportunity to do so. So we refer to that, the fact that sodium carries a positive charge and the inside of the cell is negatively charged, right? That's the fact that sodium is attracted to that negative charge inside the cell. It constitutes the electro part of the electrochemical gradient. Now, the chemical part of the electrochemical gradient just refers to the fact that sodium has a higher concentration outside the cell than inside the cell. And as a consequence of the way that concentration gradients work, right, sodium will move from areas of high concentration to areas of low concentration along a concentration gradient if it's given an opportunity to do that. That's the chemical part of the electrochemical gradient. So what the sodium-potassium pump does is it establishes electrochemical gradients. This electrochemical gradient for sodium is particularly important. Same with potassium, right? Now, predominantly, the movement of potassium out of the cell is the chemical gradient, but we'll talk about that when we get into the Nernst equation. So think of sodium for the time being. Now, how do we use the electrochemical gradient? What do we use the electrochemical gradient for? Well, one of the things that we use the electrochemical gradient for is we do work with it. So secondary active transport, the maintenance of water balance in the cell, endocrine sensing, and then ultimately the transmission of electrical signals is dependent on the electrochemical gradient. So we're going to go over each one of these things one by one. Secondary active transport, there's a like fruit fly flying in front of my face. Get out of my face, man. So secondary active transport does not use ATP directly. So here we have primary active transport, and we're looking at the sodium-potassium pump. For every cycle of the sodium-potassium pump, 3 sodium ions are moved into the ECF, 2 potassium ions are moved into the ICF at the expense of 1 molecule of ATP. Sodium then builds up outside of the cell, and that sodium outside of the cell is being attracted to the inside of the cell by two things, the electrical and the chemical gradient. So when you talk about the electrochemical gradient, we're really talking about, in this case, the gradient for sodium. Now, sodium, because there's such a strong energetic pull to move sodium back into the cell, we can use sodium to do work. We can use that electrochemical gradient, the gradient for sodium, to do work. One of the pieces of work that we use it to do is to facilitate what's called secondary active transport. So any transport process in which solutes are being moved against their concentration gradient, not with, but against their concentration gradient, in which ATP isn't being used directly, but the electrochemical gradient that was established by a primary active transport process is being used, in other words, sodium is being used, is what's called secondary active transport. To put that in other terms, whenever you see the movement of sodium coupled to the movement of something else, whether they're going in the same or in opposite directions, you are looking at secondary active transport. There's no reason to use that sodium gradient to move solute particles where they're going to go anyway. So the movement of sodium is coupled to the movement of another solute particle, and that physically provides the energetic kick to move, let's say, glucose with the sodium glucose cotransporter against its concentration gradient, the glucose concentration gradient. So now we've used the energy from sodium to move something else, and that's called secondary active transport. So whenever you see the movement of sodium coupled to any other solute particle, secondary active transport. And you can think of it analogous as trying to get into a club. So let's say there's 50 dudes waiting outside of a club, and they can only get in if they're accompanied by somebody that identifies as being female. Then that female is kind of like that sodium, and that's what provides the energetic kick to move those dudes against their concentration gradient, right? From areas of low dude to high dude, outside the club to inside the club. So in that example, sodium would be like the female facilitating that, providing the energetic kick, and glucose would be like the male, just pathetically being dragged along. So, secondary active transport. Whenever you see the movement of sodium coupled to the movement of any other ion, we are talking about secondary active transport because at that point the electrochemical gradient is being used. Antiporters move solute particles in opposite directions. So in this case, the energy from sodium is being used to move calcium in the opposite direction. You're going to notice that calcium always has a higher concentration outside the cell than inside the cell as a consequence of this secondary active transport process. Or you can have symporters, which physically, move both sodium and whatever other solute you're trying to move in the same direction, like the sodium glucose cotransporter or sodium amino acid cotransporters. But all of these are secondary active transport because the movement of one solute is being coupled to the energetic gradient inherent to sodium. Now, when you look at a graph like this, it starts to make a lot more sense when you put it into the context of the sodium-potassium pump, right? It just makes so much more sense when you think about it from the perspective of the sodium-potassium pump. So here we have our intracellular fluid, which is the fluid inside of the cells in your body. And here we have interstitial fluid and blood plasma. Both of these are forms of extracellular fluid. So when you look, for example, at the concentration of potassium inside the cell, the concentration of potassium inside the cell is very high. Why would the concentration of potassium inside the cell be very high? Well, the cell is using energy to move potassium in. Now, if you look at the concentration of both sodium and chloride inside the cell, they're very low. Well, why is that? Because we're actively moving sodium out. But if you look at the extracellular fluids, the concentration of sodium chloride is really high, whereas the concentration of potassium is very low. Now, another thing I want to highlight, the reason that I put PNEG, is protein concentrations inside the cell are very high, but in the interstitial fluid, they're relatively low. And this accounts for a lot of that charge difference you see. There are also other anions like bicarbonate ions and phosphate anions and other things, but I just put that there just to emphasize that proteins are going to have a much higher concentration. We'll discuss why you see that difference between the plasma and the interstitial fluid in A&P2. So there we go. That makes a lot more sense why you get this ionic disequilibrium. And notice that the concentration of solute particles essentially across the board, potassium is really the only solute that has a higher concentration in the cell than out of the cell that we care about for this class with respect to our inorganic ions. Now, when you look at transport processes that involve the use of a protein, and specifically carrier-mediated processes, tend to follow what's called a saturation curve. And essentially what's happening here, if you look at transport rate in units per time, and then you're looking at the concentration of that solute, as the concentration of that solute increases, the transport of that solute into the cell also increases, but there comes a point at which all of those transport proteins, so let's say we're talking about sodium glucose cotransporters, they work at a finite rate and they can only move a certain number of particles per unit time. So there's going to be a point at which you increase the concentration of glucose and sodium to an extent that all of these proteins are going to be tied up transporting glucose and sodium, and there's not going to be any extra ones. So the glucose and sodium that are backed up are going to have to wait in line. And the point at which all of those proteins become tied up, where it doesn't matter if you increase the concentration because the transport rate will remain the same, is called a transport maximum. And if you want to think about transport maximums, think about, let's say we have 3 bank tellers. And each of those bank tellers takes 5 minutes to handle a customer. So you have one customer, that customer will get in and out in 5 minutes. You have 2 customers, right? So you have one customer will get in and out in 5 minutes. You have 2 customers, so let's say you increase the concentration here, you increase the number of customers. You'll now move 2 customers in that same period of time. So you'll move 2 customers, right? 2 customers will get out in 5 minutes. If you put in 3 customers will get out in 5 minutes. But if you have 4 customers waiting, right, the max, or 6 or 10 or 20 customers waiting, the maximum rate at which those tellers can work is still just 3 customers for every 5 minutes because they can't move any quicker, and it doesn't matter how many customers are waiting in line. It's kind of analogous to what you see here. Now, I always like to draw that out, but I don't have a little pad and I'm in a hurry. Now, when we think about transport processes, right, like primary active transport, secondary active transport, they're really important to an aspect of medicine called pharmacokinetics. And pharmacokinetics essentially it's where drugs distribute in the body and how they exit the body. So when you take a medication, how does it distribute in your body and how do you get rid of it? And there's a saying for there's a mnemonic to learn the basics of pharmacokinetics. It's called ADME. So you take a medication, how is it going to be absorbed? How is it going to be distributed in the body? Well, that distribution is largely dependent on transporters. How do we get a medication from the outside to the inside of a cell? Once that medication is distributed, because you don't want it building up in your body, how is it metabolized? Is it broken down in the liver? Is it broken down in the cell directly? And then how does your body get rid of it? And this is a really complex set of concepts, but it relates directly to transport processes. So I'll give you a real-world example. Now, you're going to see diagrams like this a lot in A&P2. And what you're looking at right here in this top right-hand corner is a little structure in your kidneys called a nephron. Now, this nephron has a tube we refer to as the renal tubule, and when you filter blood and that plasma gets pushed into this little tubule, that plasma becomes what's called filtrate. That filtrate gets modified as it moves through the nephron, and any of that filtrate that remains in the nephron as it exits your kidney essentially becomes urine, and you physically get rid of that stuff in your urine. Now, you have substances in your blood So here we have blood over here, and this OC with a positive represents what's called an organic cation. So we have this organic cation over here. Now, about 40% of the medications you take exist as organic cations when they're dissolved in your body's extracellular fluid, like your blood. So you can think of this as being like a medication or maybe even a toxin. Now, that organic cation, because the body wants to get rid of it, it wants to move it from the blood into the actual lumen of the tubule so it can be swept in the filtrate and ultimately eliminated in the urine. In other words, whatever we move over from here in the blood to the lumen is ultimately going to be eliminated in the urine. So this organic cation binds to a transporter called the human organic cation 2 transporter, which physically moves it from the blood into the cell. And then it binds to these proteins, mate 1 and mate 2, which are the multi-drug and toxin extruder proteins, which physically move that cation from the inside of the cell into the lumen, where it's going to be eliminated in the urine. And you can think of that as being a toxin. So we pay attention to these transport processes all the time because they're really important with things with respect to how drugs, for example, distribute through the body. Now, if we were to throw in, let's de-abstract it, and let's say that we're putting in metformin. Metformin is an incredibly useful medication for treating type 2 diabetes. It increases insulin sensitivity and decreases gluconeogenesis in the liver. It's really important. It lowers glucose levels. So metformin is eliminated via this particular pathway. Not hypothetically, it is. That's how metformin is eliminated in the body, and then you urinate it out. So it's not really hard on the liver because it isn't getting metabolized by the liver. It's being eliminated in the urine by the renal system. Now, metformin lowers blood glucose levels, but somebody who's diabetic who needs to take metformin or who just wants to live longer, if you enjoy watching different researchers who study longevity that potentially take a compound like this, but whatever. Metformin, right, is not the only compound that this particular transporter can move. These multi-drug and toxin, these organic cation transporters and organic anion transporters are kind of like garbage disposals. They're unique in the fact that they can bind to multiple substrates and move them. So let's say we introduced another medication like cimetidine. And cimetidine is a medication used to treat gastroesophageal reflux disease, which is really common in diabetic patients. Well, cimetidine binds to and competes with metformin, meaning that it takes up that space that metformin would usually take up on that protein, and it slows the movement of metformin from the blood into the urine. If you're taking both of those medications together, metformin will start to build up in the blood, which could drop your blood glucose levels dangerously low and could result in something like passing out behind the wheel. What you are looking at here is one potential mechanism of a drug-drug interaction. Now, most of those drug-drug interactions happen in the liver, but drug-drug interactions also happen in the kidneys. And what you're looking at is just a functional consequence of transport kinetics. You're looking at transporters and what they do, and if you disrupt what they do, you can have massive effects on a patient's health. So, another thing the sodium-potassium pump does is it maintains water balance in the cell. How does it maintain water balance? Well, we talked about one of the rules with respect to the way that water moves. Water will always follow solute, and specifically non-penetrant solute. So if you're moving 3 sodium ions out of the cell and only 2 potassium ions back into the cell, you will be moving net water out of the cell, which will prevent the cell from swelling and lysing. So it's a way to, by moving net solute out of the cell rather than into the cell, that's a way to actually drag water out as well. Endocrine sensing is just using the gradient. So I won't go into detail here because we haven't talked about this enough, but it's using the gradient to allow cells to sense what's going on in their environment. And those can be the cues, for example, for releasing chemical signaling molecules such as insulin. We use that electrochemical gradient, that membrane potential, to do things like this. So another discussion we're going to have is, and this is getting into our final set of key points, so this is key point 3. We've talked about primary active transport. We've focused on the sodium potassium pump. We've talked about the generation of the electrochemical gradient. Now, when we talk about my phone's ringing, sorry. When we talk about neurons, oh, stop, bro. Are you gonna, no, man. So when we talk about neurons, we're also going to talk about the role of what are called potassium leak channels in establishing the membrane potential, and we're going to talk about the negatively charged particles inside the cell. Potassium leak channels are really important. As we look at this, oh, stop. Okay, as we look at this, we have key terms to recognize here. So there are a few different types of channels. One type of channel, which is really a receptor, right? And remember, receptors are proteins that mediate communication between the outside and the inside of the cell, is what's called a ligand or a ligand-gated ion channel, depending on who you take. So a ligand is a blanket term for a chemical signaling molecule, like a hormone or a neurotransmitter. And that hormone or neurotransmitter comes here and it binds to this channel protein. Now, before that hormone or neurotransmitter, that ligand binds to this channel protein is closed. After that ligand binds, right, these are also called chemically gated ion channels, it triggers a change in that protein shape that physically opens it up and allows for the movement of things like sodium, chloride, calcium, potassium. And we would refer to those as being things like ligand or chemically gated sodium channels, chemically gated potassium channels, or ligand gated sodium channels, ligand gated potassium channels. So those require a chemical messenger to open. These are really receptors. They're a type of a receptor. Voltage-gated channels, on the other hand, notice that it's negative in the ICF and positive in the ECF. Voltage-gated ion channels are forced to open when charge inside the cell changes, which forces the protein to move in a certain way and go from a closed to an open state. So ligand-gated ion channels are found in certain areas of a neuron. Voltage-gated ion channels are found in different areas of a neuron. Specifically, ligand-gated are found in dendrites and voltage-gated are found along the axon. Another type of ion channel is what's called a tension-gated or a stretch-gated ion channel. In this case, mechanical force, like if you touch and trigger a touch receptor, it's mechanical force that physically opens up these channels and triggers the depolarizing. Hey there, bad editing. I don't care. So, the final thing we're going to talk about is the difference between what's called depolarization and hyperpolarization, because these are really important concepts to understand with respect to membrane potential. So if you look at this cell over here, capital letters or big letters mean that you have a high concentration of a solute. So potassium has a really high concentration in the cell. Sodium chloride, on the other hand, has a really high concentration outside the cell. In fact, the only inorganic ion that has a higher concentration inside the cell than outside the cell is potassium. If it's any other ion slash electrolyte that we're looking at, you can assume that the concentration outside of the cell is going to be higher than that in the cell. And I could give you specific numbers, but that's not the focus of what we're doing here. So a lot of anatomy and physiology instructors will ask a pretty straightforward question. What do you think changing the permeability of a membrane to a specific ion will do to that cell's membrane potential? Will it make that membrane potential more positive? Will it make that membrane potential more negative? So remember, resting membrane potential is negative 70 millivolts. Anything that drives resting membrane potential below negative 70 millivolts to like negative 75, negative 80 millivolts is considered what's called a hyperpolarizing shift, because the charge difference between the inside and the outside of the cell is getting greater, right? So its polarization is decreasing. Any electrical shift in which the cell becomes more positive, right? So you go from, let's say, negative 70 to negative 60, that's considered a polarizing shift. Now, hyperpolarizing shifts, especially when we're talking about neurons, are always inhibitory. They're always inhibitory. Depolarizing shifts are always excitatory. And this establishes a framework to think about a lot of the neurophysiology that we're going to be talking about later on in the class. So let's think about what that actually means. When you say increase the membrane permeability to an ion, what you're really talking about is opening up a channel. When you say decrease the membrane permeability to an ion, what you're really talking about is closing a protein channel. So if I said if the membrane permeability to for potassium is going to increase. So the question writes, which of the following would be true if the membrane permeability for potassium increases? The first thing we have to think about is what does membrane permeability mean in biological systems with respect to ion permeability? The only thing that can increase the permeability of potassium to the membrane is the opening of potassium channels. So I'm asking you, which of the following would be true? Now, if potassium channels open, let's say potassium channels open, in which direction is potassium going to move, assuming that the sodium-potassium pump is working and that the concentration of potassium is higher inside the cell than outside the cell? Well, potassium is going to move from the inside of the cell to the outside of the cell. And what does potassium carry, a negative or a positive charge? Well, it carries a positive charge, so positively charged ions leaving the cell, right, is going to decrease the charge in the cell because there's going to be more of those fixed negatively charged ions. So when potassium leaves the cell, what's going to happen to the cell is it's going to become more negative. Now, one of the factors that plays a really important role in establishing the membrane potential of a cell, that negative charge inside the cell, right, is the sodium-potassium pump. Another are what are called potassium leak channels. And those potassium leak channels are just open all the time. And what they do is they allow potassium to exit the cell, which takes the membrane potential more negative, closer to what's called potassium's equilibrium potential, which is something, again, we'll talk about when we talk about the Nernst equation. Now, assuming the cell is at negative 70 millivolts, the sodium-potassium pump is working, potassium's exiting out of those potassium channels, and then we open up more potassium channels, what's going to happen is that cell is going to become more negative than negative 70. And when the cell becomes more negative than negative 70, like negative 75, negative 80, we say that that cell is hyperpolarized. And we refer to hyperpolarizing shifts, especially when we're talking about neurons, as being inhibitory. That's a way to turn a neuron off, is to make it even more negative. Now, let's say I was to say we increase the membrane permeability to sodium. Well, sodium, assuming the sodium-potassium pump is working, which you always do in living systems, has a higher concentration outside the cell than inside the cell as a consequence of the sodium-potassium pump. So if we increase the membrane permeability to sodium, what we're really saying is we're opening up a sodium channel, or we're opening up sodium channels. And if you were to open up a sodium channel, in which direction is sodium going to move? Well, if you open up sodium channels and you increase the permeability of the membrane to sodium through doing that, sodium is going to move from the outside of the cell to the inside of the cell. And what type of charge does sodium carry? It carries a positive charge. So if millions of little sodium ions are rushing into the cell isn't going to become more negative. The cell is going to become more positive. So you're going to see a shift where the cell goes from being like negative 70 to negative 65 or negative 60, right? Or any other number, depending on the amount of sodium that rushes into the cell, as long as it's more positive than negative 70. When the cell becomes more positive, we say that the cell has depolarized because the electrical potential inside the cell becomes more similar or less polar to the electrical potential outside the cell. So sodium rushing into the cell depolarizes the cell. Now let's say we had chloride. Chloride also has a higher concentration outside the cell than inside the cell. Let's say that we increase the membrane permeability to chloride. So we open up a chloride channel. That's another way of saying that. If we open up that chloride channel, chloride is going to rush from the outside to the inside of the cell. And what type of charge does chloride carry? Is it going to depolarize or hyperpolarize that cell? And remember that all ions, with the exception of potassium, have higher concentrate. All inorganic ions that we care about in this class, right, uh, have a higher concentration inside the cell than outside of the cell. But pardon me, have a higher concentration outside the cell than inside of the cell. So sodium, chloride, calcium, magnesium have higher concentrations outside of the cell. The only ion with a higher concentration inside the cell, inorganic ion, is potassium. So that chloride is going to rush in, and what's going to happen to the cell? It's going to become more negative. So think about the image you're looking at here. How would you characterize this shift? Well, is this cell, right, becoming more positive, or is it becoming more negative? If it's becoming more positive, you would refer to this as being a depolarizing shift. If it's becoming more negative, it would be a hyperpolarizing shift. What ions could cause that to happen? Well, think about the fact that all ions have a higher concentration outside the cell than inside the cell, with the exception of potassium. And think about the movement of those ions. Are those ions going to cause the cell to depolarize or hyperpolarize? Are they going to cause the cell to become more positive based on their movement, or more negative? And what ions could account for this? It's not just one. So think about that in your questions. On the flip token, what happens when the cell becomes more negative than negative 70? What type of shift is that in membrane potential? Is it depolarizing or hyperpolarizing? What changing the permeability to what ions could cause this? What ions could actually physically cause this? And they don't always have to be rushing into the cell. Sometimes they could be rushing out of the cell. And whenever you see this shift, would it be considered excitatory or inhibitory? Right? Really think about those questions. Depolarizing shifts are excitatory, hyperpolarizing shifts are inhibitory. And as we move into talking about neurophysiology, when you look at things like the action potential, right, the electrical signal that travels along the axon of a neuron, it's all predicated upon an understanding of the sodium potassium pump and the movement of ions, and the idea of depolarizing, hyperpolarizing, and we will eventually introduce the term repolarizing. So we're talking about the movement of ions. And that should give you the springboard or the kick that you need to get that handout done and to address this first quiz that you're going to have. Thank you so much. Bye-bye.